Thank you for the opportunity to present our work on RMD 1101, an investigational first-in-class, potentially disease-modifying gene therapy for the treatment of osteoarthritis. Disclosures are at the bottom. We're all too familiar with OA, a disease that impacts several hundred million people worldwide with no currently approved disease-modifying treatments, despite a plethora of therapeutic classes investigated ranging from anti-inflammatories to senolytics. Recent advances with anabolics appear promising, with a placebo-controlled trial of FGF18 demonstrating dose-dependent increases in cartilage and even a reduction in the rate of progression to joint replacement in the high-risk subgroup. Pharmacokinetics do limit that treatment, requiring up to 12 injections in bilateral knee OA that may need to be sustained indefinitely to prevent reversion to cartilage loss. Remedium is developing an FGF18 gene therapy, and the goal of these studies was to confirm the mechanism and assess the safety and efficacy potential of AAV2 FGF18. To do this, we designed several studies to assess the site compatibility, transducibility, effect on the chondrocyte transcriptome, but ultimately in vitro and in vivo chondrogenesis. Studies with primary human chondrocytes in culture demonstrated excellent cytocompatibility of AAV2 with MOIs dosed up to 50,000. At the same time, chondrocytes showed excellent permissivity with a fraction of GFP-positive cells reaching over 80% just 7 days post-dosing at MOIs as low as 1,000. Vectors were equally cytocompatible with chondrocytes and synoviocytes. You can see this from the lack of a statistically significant dose-response curve in the top two panels. Synoviocytes did, however, appear nearly 500 times more difficult to transduce than chondrocytes, as evidenced by the MOIs required to attain the same GFP-positive cell count in the bottom panels. The ability of FGF18 to induce chondrocyte proliferation is well known. Here we show that both the full-length, fully glycosylated protein and the bacterial truncated analog are able to induce chondrocyte proliferation in a similar dose-dependent manner, but most importantly, that the same effects can be obtained by dosing with relatively low quantities of AAV2 FGF18. Both the gene therapy and protein-treated cells significantly upregulated hyaline cartilage-associated COL2 and HAS2, while reducing expression of fibrocartilage-associated COL1 and LOX. Interestingly, even at an MOI of 10, the gene therapy-treated cells were able to upregulate mucin and SOX9, while reducing expression of OA-associated ADAMS15. The RNA-seq studies suggest that FGF18 works by both the induction of chondroanabolic activity and the reduction of catabolism via downregulation of genes such as MMPs, ADAMS, and LOX. In parallel, that FGF18 appears to suppress the formation of scar-like fibrocartilage, which has been previously associated with low durability of microfracture procedures. The mechanistic studies enabled dose selection for two subsequent in vivo studies in healthy and DMM-induced OA spragg rats, which were followed for 2 and 1.5 months respectively. We first confirmed widespread transduction of chondrocytes with AAV2, which augmented naturally expressed FGF18 in the joint. The pattern of expression, depth of penetration, and lack of overt immune response were similar to previously published studies. We then confirmed stable and localized signal within the joint using bioluminescence imaging in both healthy and diseased joints, with the latter followed for up to 24 weeks, displaying no loss of expression over the study period. Interestingly, we observed significant joint swelling in the repeat FGF18 protein injection arm that was not present in the AAV2 GFP or AAV2 FGF18 treated joints, indicating that the swelling is likely caused by the continuous disruption of the joint capsule due to the repeat nature of the protein injections. The swelling did partially resolve by month 2, but was still statistically higher than the untreated baseline. Using this OARSI measure of synovitis, we confirmed that the repeat injections is associated with increased synovial inflammation. Our initial indications of chondrogenesis came from the healthy animals, where we observed increases in cartilage in the protein and gene therapy-treated groups. These increases were, however, quite modest and observed only after normalization by weight, which was significantly correlated with cartilage position. In the induced OA model, however, the differences between the control and treatment groups were significantly more pronounced. We observed a dose response from the gene therapy, shown here in blue, relative to the vehicle, shown in red, reducing both the defect width and depth, with the effects being markedly obvious from the histological sections. The overall cartilage thickness was also increased and confirmed dose-dependent activity, and these effects were observed regardless of whether the treatments were normalized by weight or the contralateral sham. Ultimately, a picture is worth 1,000 words, so the top row are all the vehicle-treated joints. You can clearly see severe cartilage erosion, with some sections showing bone marrow lesions, subchondral bone remodeling, and in some cases even complete subchondral bone collapse. The gene therapy treated joints in the bottom row look a lot more phenotypically normal, with only regional gap loss and occasional marginal cartilage erosion. In summary, FGF18 gene therapy and protein appear to act by similar mechanisms, promoting chondrocyte proliferation and highlighting cartilage-associated gene expression. Ultimately, intra-articular gene delivery of FGF18 appears safe and well-tolerated, producing marked chondroprotective and regenerative benefits. Further studies will focus on long-term safety, durability, IND-enabling pharmacology, followed by the advancement of RMD 1101 to human and companion animal clinical studies. We thank you for your time, and of course a colossal thanks to the team that made this possible, including Dr. Hollander, Dr. Zhang, Dr. McCallenden from Tufts University. Thank you. Yep, so both great questions. The answer to the first one is simple. Yes, we did. We used an anti-FGF18 antibody which showed increased expression in both the chondrocytes and the synoviocytes in vivo. While the in vitro transduction in chondrocytes was simpler than synoviocytes, actually by 500 times, that wasn't the case in vivo, and that's likely due to the extracellular matrix that surrounds those cells. There have been other groups that also published work using GFP as a reporter, a transgene that was expressed and visualized directly by fluorescent microscopy, and they observed something similar. Chondrocytes are transduced in the superficial and medial layers of the cartilage. Synoviocytes are transduced also in the surrounding area of the injection. Now, regarding your second question, and just to be clear, the repeat injections were only in the protein-treated animals. And the protein-treated animals mimicked the previous studies of FGF18, which were used to advance that protein into the clinical studies. The clinical studies obviously use, you're probably familiar, a regimen of 3 weekly injections, followed by either dosing every 6 months or every 12 months, and these need to be continued indefinitely. This is the forward trial continued them, I believe, for a period of 2 years, and then with a 3-year follow-up. The reason why those protein injections are necessary to be repeated, both in the animal models and in the clinical trials, is because of simply the washout of FGF18. It's a pharmacokinetic spike that's then reduced, another pharmacokinetic spike and then reduced. There were early studies with FGF18 in vitro that suggested that a hit-and-run mechanism for FGF18 is necessary to induce the anabolic effect. I should correct myself, rather than necessary, induce the higher chondroanabolic effect than continuous dosing. We do not observe that from our in vivo studies, so we see that even a relatively continuous expression of FGF18 produces more chondroprotective and regenerative benefits. And actually, the original study that discovered FGF18's chondroanabolic mechanism, which used an adenovirus to deliver the FGF18 transgene, observed the same continuous expression and seems to work in vivo. Whether that's a difference between in vitro and in vivo, or whether in vivo we're truly getting some fluctuation, whether it's circadian rhythms or just normal diurnal cycles of activity and rest, we don't know. But we know that in vivo it seems to work. So it's a great question.
So the point actually I'm interested in is that how long these AAVs persist inside the cartridge versus synovium.
Yeah, so we measured up to only 1 month. Yeah, we measured them up to 24 weeks using bioluminescence imaging, and you can see the expression is highly durable. There have been other groups that have taken this out to, I believe, over a year, and the durability is still the same. So they see no loss of expression, no loss of the signal over a long duration. So, yeah, another great question. Thank you.
Perhaps just one last question.
Thank you for showing a new AAV approach. And I can confirm, yes, 1 year we have observed that intraarticularly. You have shown the SAFO staining in vivo. Do you have data on the collagen and hypertrophy?
Yep, so great question. Interestingly, collagen 1 we see suppressed by RNA-seq, collagen 2 we see upregulated by RNA-seq. We're now conducting IHC studies, and that's certainly going to be part of our application. So collagen, lubricin, Col1 to Col2 ratio are something that we're going to be looking into. We haven't yet, but that's definitely on our radar to explore.
Thank you.
Okay, thank you very much.